Hello everyone. Today we're going to talk about microbes. We're going to introduce the different types of microbes. Microorganisms or microbes, those are tiny living things that are found all around us, and they're too small to be seen by the naked eye. They are ubiquitous, meaning they are everywhere. They live in water, soil, and air. Even our human body is home to trillions of these microbes. There are bacteria, viruses, protozoa, and fungi. Under fungi, we have yeast and mold. We also have microscopic algae. All of these are just examples of the different type of microbes that we have out there. Most bacteria aren't dangerous for humans. Many of them live on or inside of our body, and they actually help us to stay healthy. For instance, there are bacteria that can help us digest food in our gut, and some of them even produce vitamins like vitamin B and vitamin K. And vitamin K is actually known to help us clot our blood. Some bacteria are even used to produce food such as cheese and sauerkraut, yogurt, pickles. Some of them produce things like ethanol and acetone and even oxygen. Actually, less than 1% of all the bacteria out there are responsible for the diseases, and we refer to them as pathogenic. So the term pathogenic means disease causing. And again, not only bacteria, some bacteria out there are known to be pathogenic. We also have viruses. We are in the midst of a pandemic right now, COVID-19 virus. So viruses are also pathogenic. And so pathogenic just means the ones that would cause harm to us, disease. Some other benefits of those microorganisms is we can actually use some of them to produce things that would help us humans, for example, insulin. Now, this idea of using a bacteria to actually get a human product such as insulin is due to the fact of a technology known as recombinant DNA. And that's a technology scientists developed that made it possible to actually insert a human gene into the genetic material of a bacterium. And so we refer to that as recombinant microorganism. And that recombinant microorganism would be able to produce the protein encoded by the human gene that was inserted in it. And that's actually what they did with the insulin production to accommodate the high number of diabetics that are in need of insulin. They took the insulin human gene, they inserted it into the bacteria, and the bacteria started making the human insulin. So we see how microorganisms can actually be also used in the COVID-19 vaccine. For one of the vaccines that are now, got developed, they actually went ahead and they took a piece of the COVID virus, they inserted it into an adenovirus, which is a common cold virus. And so when they inject it eventually into the host, when they inject that adenovirus into the host is going to start making antibodies against the COVID-19 virus. So as you can see, having knowledge of these microorganisms can actually benefit us. We can take advantage, as I said, of inserting genes from different organisms into bacteria or into viruses so that we can start making products like insulin, or we can start making antibodies. So it's definitely helpful for the pharmaceutical companies, for the health industry, Even for the food industry as well, like if we have knowledge about those microorganisms, we're going to be better off in preventing food spoilage. Now, the microbiota, we refer basically to the microbes that live inside of us or on our skin as microbiota or microbiome. And this is an image right here of the microbiota that you would actually find on the surface of your tongue. So basically what you're seeing, all of these are different types of bacteria that you could actually find on your tongue. And so actually this microbiota that you are seeing right here actually help us maintain good health. Without it, we could actually start falling ill. But the next time you share a glass of water with someone, you're not just sharing water, you're also sharing your microbiota with them. Let's talk about nomenclature, meaning how we name those microorganisms. The scientist that came up with this system is Carolus Linnaeus. He came up with that naming system and he established that scientific nomenclature in 1735. Each organism basically gets two parts for the name, the genus and the species. Now, the way we write the microorganism name, the name would be italicized. For example, right here. You would see that this bacteria right here, Escherichia coli, it's actually italicized. This would be the genus and this would be the species. So Escherichia would be the genus, coli would be the species. The genus, the first letter of the genus name always gets capitalized. And that's it, only the first letter. Now, the species never gets capitalized. So you could write it as follows, or instead of writing the whole entire genus name, you would just abbreviate it by just putting the first letter. So it would be E. coli. You never abbreviate the species. So only abbreviate the genus, but never abbreviate the species, or you can write it out fully like so. So this is how we go ahead and we write the names of the various bacteria, for example, such as Escherichia coli. And so, as I said, it's usually italicized. Now, if you cannot italicize it, then you would underline it. So you would underline the entire name. And as I said, the genus first letter would get capitalized, and then the species would be lowercase. The names of the microorganisms are actually in Latin because that's what they used back then. So all the names are in Latin, and the names usually represent something. So either the name is honoring the scientist who actually discovered that microorganism, or the name is actually telling you something about where that microorganism resides. So an example, if we take a look at the name of this bacteria right here, E. coli, now as I said, when you come and write it, this would be the genus, this is the species, either italicize the whole entire name or underline the genus and the species. Never capitalize the species, and for the genus, capitalize the first letter. Escherichia coli, the coli portion is actually giving you a hint about where would this bacteria be located inside of the human. So inside of a human, this bacteria would actually reside in your colon, which is your large intestine, and hence the name coli. So as you can see, the names sometimes of these microorganisms can give you hints about where they are located. And then also the genus portion of it, it's honoring the scientist that actually discovered E. coli. Another example, we're going to take a look at Staphylococcus aureus. The genus Staphylococcus, we're going to break it down into two parts, Staphylo and coccus. Staphylo actually means clusters. So you can see that this bacteria, when you take a look at it under the microscope, their cells would actually be clustered together, and that's what Staphylo means. Coccus means spherical. So you can see that the bacteria are spherical shaped. Aureus actually means gold colored. So it has that golden color to it. And so you can see that this name right here, Staphylococcus aureus, gave you information about the arrangement of the bacteria. So it's arranged in clusters, the shape of the bacteria, which is coccus, spherical, and the color, aureus, which is golden. And Staphylococcus aureus is a bacteria that's found on your skin. Now, we have different types. We have different types of microorganisms, and we'll go ahead and take a look at each one of those and talk a bit more about each one. So let's first talk about bacteria. So bacteria, they are considered prokaryotes. The prokaryotes is actually a domain, so they are under the domain known as prokaryotes. Pro actually means before. So prokaryote actually means before a nucleus, before the development, or before a nucleus was originated. And the reason why bacteria are prokaryotes, because they do not have a nucleus. So that's why they are under the group of prokaryotes. They don't have a nucleus. Bacteria, they are very simple. They are single-celled, so only one cell. They do have cell walls, and their cell walls are actually made out of carbs and proteins, and their cell walls are made out of peptidoglycan. So peptidoglycan, it's a common feature in bacteria. Not all microorganisms have peptidoglycan cell walls. That's a feature for bacteria. And bacteria, they divide via binary fission, which actually just means that this bacteria right here, when it wants to reproduce and make more of itself, all it does is it goes ahead and it splits itself in half, and you get two cells. And that process is actually pretty quick. Within 20 minutes, you can actually have a whole new, colonies, not just one colony, but like colonies and colonies of bacteria, all in just 20 minutes. So it's a pretty fast process. Bacteria, they do need nutrition. So if you do want to grow bacteria, you have to provide them with nutrition. Otherwise, they're not gonna do very well. They can get their nutrition from either organic sources. Whenever you see the word organic, it means that the source that you're talking about has a bunch of carbon elements in it. So that's what organic sources are mostly made out of, like lots and lots of carbons. So they can, get their energy from eating up organic stuff or inorganic chemicals. The end of the day, bacteria need nutrition. Now, later in the semester, we're going to discuss how some bacteria, they are a bit more picky, and so they need special, like, nutrients. Some bacteria, they can survive, on anything, okay, so they are not very picky. And so some bacteria also, another thing to mention is some bacteria are actually known as photosynthetic bacteria. An example is cyanobacteria, and that means that they get their energy through photosynthesis. Next, let's discuss archaea. So archaea, they're also considered prokaryotes. They do have cell walls, but their cell walls don't have peptidoglycan. Um, they're they're very similar to bacteria, but archaea, they usually live in very harsh conditions. So they love to live in such extreme conditions. For example, they love to either live in a very salty environment, and so they thrive in high salt concentrations, and so we refer to them as halophiles. Some of them, they love to live in like super hot conditions, like near a volcano or hot springs where it's extremely hot, and so we refer to them as thermophiles. Um, and some of them love to live in very cold conditions. Some of them, we refer to them as methanogens, and those are the ones that usually produce methane gas, which is a gas that has a pretty foul odor to it. Not a lot of, actually, I should say, we didn't till now find any archaea that are pathogenic, disease-causing archaea, and that's why we don't give too much attention to them, because they don't really harm us. So most of the time you're going to find that scientists are more concerned about learning about microorganisms that either harm us, so we can understand them better, or, if they are going to benefit us in a way. So that's why we're not going to talk too much about archaea in this class. Let's talk about fungi. Fungi, they are eukaryotes. They do have a nucleus. So far we talked about bacteria and archaea, and those are prokaryotes. Fungi are eukaryotes. They have a nucleus, they have cell walls. Their cell walls have chitin within them. Chitin is a different component compared to peptidoglycan that's usually found in bacteria cell walls. Fungi, they do need energy so that they can thrive. They can use up organic chemicals. Some examples of fungi out there are yeast. Yeast are actually unicellular, meaning they're only made up of one cell. Some yeast out there can actually cause infections to humans. So when you hear someone having a yeast infection. And then, of course, yeast is known to be used in bakery, right? So we use it to go ahead and make bread. And we use yeast to also make wine. Other examples of fungi, we've got molds and mold and mushroom. Mold and mushroom, those are multicellular, so they do have multiple cells. An example of mold is when you find it, for example, on a piece of spoiled bread, or spoiled food that has gone bad, and when you see that fuzzy, furry stuff growing on top of your bread, for example, that's basically mold. And that furry, fuzzy stuff that you're seeing, we refer to it as mycelia. So mycelia is basically that hairy stuff that you're seeing on your spoiled food. Um, Mycelia is basically a bunch of filaments known as hyphae. And we will talk more about it in detail later when we discuss fungi together. Protozoa are eukaryotes. Examples of protozoa would be amoeba, and that's actually an image right here of an amoeba. Amoeba, you would find it, for example, in ponds. Okay, so amoeba, they are microscopic. You won't see them with your naked eye, but they are usually microscopic organisms that live in ponds. Not all protozoa live in fresh water, so some of them also live in salty water as well. And you can see right here, this amoeba has like these kind of extensions coming out of it, and those we refer to them as pseudopods. Pseudo means fake, pods mean feet, so they kind of look like they have fake feet. And they use those pseudopods to basically crawl on surfaces. Some other protozoa out there, they have cilia as a way of movement. So um, cilia are basically tiny hair-like structures that would help the protozoa move. Flagella is another way to help with movement. So flagella, it's a whip-like structure, so it can help the protozoa move. Again, protozoa, they are either free-living, they live on their own, or they are parasitic, meaning they do need a host. So they need to find the host so that they can use the host's resources so that they can survive. Another group of eukaryotic microorganisms are algae. Algae, they are eukaryotes, they do have cell walls. Their cell walls contain cellulose. You do find algae either in fresh water or in salty water. Even some algae are actually found in soil. Some of them are actually photosynthetic, so they use photosynthesis to get their energy. And especially the photosynthetic algae can actually produce oxygen. So they can actually produce oxygen, and that's just a picture right here of some of the algae that you might actually find in pond water. Viruses are neither prokaryotes or eukaryotes. They are a group on their own. They don't have any cells, and we refer to that as acellular. Inside of a virus, you would either find DNA or RNA. And surrounding that nucleic acid, there is a protein coat that's protecting that DNA or RNA. And some viruses, they actually have a lipid envelope protecting their protein coat. So they get like an extra cover. And viruses, we're actually going to talk about how viruses attack your cells and invade your cells because they could actually invade it in multiple ways. Viruses, they definitely need a host. They're not going to be able to survive for a very long time on their own. So they're always on the hunt for a host. And humans, we are such good hosts for them, and they get to use all of our resources. And that benefits them, but it definitely harms us. So viruses, they definitely need hosts, and that's why some scientists believe that viruses are not alive because they cannot survive on their own. Even though once they are inside of the host, they would replicate and they would use the resources and everything. Again, there is a debate amongst scientists whether to consider viruses alive or not. Example of multicellular parasites would be worms. We actually have different type of worms. Some are flat, some are round worms. And worms in general, we refer to them as helminths. We are going to go in detail about exactly what we call them and the different types of worms out there. If you're wondering, why are we even talking about worms since we do see them, without a microscope, so why consider them a microscopic organism? Well, because during their life cycle, especially at the very early stages, they actually have microscopic stages, and you would need a microscope to view these stages. Now, once they mature, yes, you don't need a microscope to see these worms, but that's why we go ahead and we discuss them in microbiology as well, since they do have microscopic early life stages. Now, when we classify the microorganisms, right, like all of what we've talked about, fungi, bacteria, archaea, etc., we can actually classify them under 3 domains. The 3 domains are the following: bacteria, archaea, and eukarya. Bacteria domain, that's where you find all your bacteria. Archaea, that's again all the different types of archaea that you have. Both bacteria and archaea, those are prokaryotes. The domain eukarya, you actually have different groups or subgroups, I should say, under the eukaryotic group. You have protists, such as amoeba, fungi, such as yeast and mold and mushroom, plants, basically all the plants out there, and then animals, and we fall under the group of animals. So all of these are basically subgroups under the group or the domain known as eukarya. And that was developed by Carl, and he was the one who classified these 3 domains. Now let's talk about some microbiologists that actually made huge discovery to the world of microbiology. So we're going to start with Robert Hooke. Robert Hooke was actually one of the first scientists that was actually able to look at cells. And back then, he actually didn't call them cells. He used to actually call them little boxes. And he was able to see those little boxes, which eventually we call them cells. And he actually came up with the cell theory. So he started the cell theory, and the cell theory is basically saying that all living things are composed of cells. Later on, another scientist came along, Anton Leeuwenhoek, and he was able to see microbes under the microscope. So Hooke was able to see the cells, but Anton was actually able to view even smaller things. And he was able to do that by modifying the microscope that they had. So he was actually able to see these microscopic organisms. And he was able to view them. And later on, of course, more advanced microscopes came about. But let me show you the microscope that Anton was actually using. this is it. So this was the microscope that he was using, which is amazing to kind of see how a microscope started off and how microscopes look now. So this is how it looked like. It was handheld, so he would just kind of hold it up and he would put the specimen and here there would be the lens. He would just kind of hold it up against his eye and he would take a look at the specimen and he was able to actually see microorganisms. Of course, now, as I said, microscopes have gone a long way and they've developed and now um, such amazing microscopes that can see so much detail. There are two theories that we're going to discuss. One is known as spontaneous generation. And spontaneous generation, that was back then when people and even scientists back then, they believed the forms of life could actually arise spontaneously from non-living matter. Um, for example, they used to believe that flies and maggots would just emerge out of nothing, like out of trash. You would have a fly, emerging out of trash, or they would just believe, for example, that again, like, flies would just come out of dead bodies. They actually believed that for a while until Louis Pasteur came and disproved that thought. And we're going to talk about how he disproved it in just a bit. But the other theory that I wanted to talk about, or the other concept, is biogenesis. And that's the hypothesis that saying that all living things arise from pre-existing living cells, okay? And so those are the two, again, like a lot of people were having so much debate whether biogenesis is correct or whether spontaneous generation was correct. But I mean, now we know spontaneous generation is definitely not true. Okay, let's talk about the golden age of microbiology. And they call it the golden age of microbiology because during those years, lots of important discoveries were actually made in the field of microbiology. For example, Louis Pasteur, he was actually a French scientist, and he was the one that discovered the process of fermentation. So how did he discover the process of fermentation? What actually happened was the French merchants, they came actually to Pasteur asking him to help them out. They told him that their wine was going bad and they were not able to sell it, by the time they ship it, it's already gone bad. And so they were losing a lot of money. So they went to Pasteur, the microbiologist, to go ahead and help them out. So as I said, to make wine, yeast is usually added, right? So what happens is when yeast is added and you have the yeast in a bottle and there is no oxygen, what's going to start happening is yeast is going to start fermenting. It's going to start using up the sugar that's found in the wine and it's going to start, turning it into alcohol. Okay, and so first of all, Pasteur, that was his first discovery, the process of fermentation. Like he went ahead and he, first of all, explained to them the process of how alcohol is made because they didn't even understand how alcohol was even being made in the first place. And so again, he explained to them that when you put yeast in anaerobic conditions, which is basically a condition without oxygen, the yeast is going to start turning the sugar into alcohol. Back then, they actually used to think that just by putting sugar out in the air, like grape juice out in the air, it's going to turn alcohol, and so they didn't think of there is actually a microorganism that's actually making the alcohol, and in this case, yeast, for example. Now, okay, but then that was his first discovery, right? The process of fermentation. But then now they wanted to know, well, okay, why is it going bad? Okay, and so he discovered that the reason why it was going bad was not due to the yeast, but actually it was due to bacteria. The bacteria in the wine was actually turning the alcohol into vinegar, and that's why it tasted bad, because it was turning into vinegar and it's like very sour, and so that's why that's what he discovered. And after discovering that, he was actually, able to help them understand why their wine was going bad. And his third discovery, so Pasteur has so many discoveries, his third one was the process of pasteurization. Hint, they called it pasteurization because of his name, Pasteur. So pasteurization, it's the process of when you take a product and you expose it to extreme heat to kill the bacteria. And once the bacteria is killed, that's basically going to prevent the food from going bad, or in their case, to prevent their wine from going bad. So he ended up telling them, okay, for your wine not to go bad, you just need to heat it so that you can basically kill all the bacteria. And so they were very grateful for him because their wine did not go bad. And now we actually use the pasteurization process, for example, to pasteurize milk. So we go ahead and we pasteurize our milk so that we can go ahead and kill most of the bacteria that's found in the milk. As I said, Pasteur had so many discoveries. Another one was he was the one that disproved the spontaneous generation. So let's see how he went ahead and he disproved that. Another important discovery was made by Petrie. And Petrie, he was the one that came up with the idea of Petri dishes. And those are the dishes that we use in microbiology labs. We put on those dishes agar, which is a gelatinous material. We get it from seaweed, and bacteria cannot eat that agar. So it actually works as a very good surface to grow the bacteria. And so that was a pretty important discovery, because that even that gave an opportunity for scientists to even grow bacteria so that they can study them. And so we call those dishes Petri dishes in honor of Petrie, who came up with that idea. Another important scientist, Lister. And you can kind of think of Listerine, and Lister was actually the one that came up with the idea of aseptic surgery. So again, the Listerine mouthwash, it helps you prevent, bacteria from growing in your mouth. And so he was the one that came up with the idea of aseptic surgery to prevent infections. And till now, we actually use his aseptic techniques, and we even use his aseptic techniques in our Another important discovery was by Robert Koch. And Robert Koch, he was the one that discovered that specific microbes can lead to specific diseases. So back then, people didn't even think or believe that microbes can cause disease. They actually used to believe that if someone got sick, that was a punishment. They sinned and therefore they're getting punished by getting sick. Or they thought that it was something paranormal, or it was, it was the demons that made people sick. Not microbes, not pathogenic, microbes. They didn't believe that. And so Koch, he discovered that no, there are microbes that can cause specific diseases. There is also Winogradsky. Winogradsky, he was actually a Russian scientist, and he was able to actually grow bacteria in a column. And some of the bacteria grew at the top of his column towards the bottom. He got different colors because bacteria could actually, color, get colored differently, and you get all of these rainbow colors actually. You can actually make a Winogradsky column. I can tell you about it later, but or you guys can look it up and see how a Winogradsky column is made so that you could just have an idea. And basically, based on the needs of the bacteria, they would grow at different parts of that column. And again, he was the one that came up with that discovery. Now, back to Pasteur. Pasteur, he was able to disprove the idea of spontaneous generation. So how did he disprove the idea of spontaneous generation? One more time, just to refresh your memory. Spontaneous generation was the idea of, living things can actually come out of non-living materials. So if you have a bag of trash, that bag of trash can go ahead and start making flies. So he, Pasteur, disproved that idea. And so how did he do it? He basically got a flask, okay, and he poured some broth inside of that flask. And then what he did was he went ahead and he started heating that flask. So when he heated the flask, any bacteria, right, any bacteria that was in the broth, he was trying to kill it. Okay, and so what happened was due to heating the broth, he basically sterilized it. And then after he went ahead and he boiled it for several minutes, he said that he's going to go ahead and bend this part right here that's connected to his flask. So he went ahead and he bent that part right there. He bent the neck basically of the flask. He didn't seal it. So he still wanted air to get in because he didn't want to seal it because people thought that air was what's causing things to go bad. And so he wanted, again, to disprove that idea. And so he left it open, but he went ahead and he bent it. And so the reason why he bent it, super smart of him, because if there are any microorganisms in the air, any bacteria, any viruses in the air, if they enter into the flask, and right here they're going to get stuck. They're going to get stuck in that bent portion. It's going to be hard for them to make their way all the way into the flask. And so he actually, by doing that, he disproved the, spontaneous generation because the broth didn't go bad. And so he told them, there you go, like, air is still getting inside, but yet the broth is sterile, and that basically disproved the spontaneous generation idea. And they actually have Louis Pasteur's flask in a museum in France. Of course, now it's sealed because it's not going to stay sterile for that long, for such a long time. But they actually have it in France, and the broth still looks pretty good. I mean, it's been now over 100 years now since Pasteur did this experiment, and the broth is not contaminated. So that's amazing. But as I said, now they do have it sealed. So again, that this proved that air can just make things go bad. Like, you have to have bacteria in the air to make the food go bad. Otherwise, it's not, right? So that was his also, another one of his important discoveries. Now Lister, we talked about it, right? So Lister, he was the one that came up with the aseptic technique, and he actually helped lots of physicians because back then they never used to wash their hands when they used to see their patients. And so lots of patients would end up dying because of the infections, and they didn't actually know why. They had no idea why patients were dying. And so Lister came up with the concept of, well, we have to wash our hands, and we have to sterilize our equipments, and we have to, sterilize everything. So he actually started using something called phenol to disinfect his surgical tools and, to disinfect the wounds. And by doing that, patients were not dying as before because they started, disinfecting everything. And that was a pretty important discovery. Robert Koch was able to link a specific bacteria to a specific disease. How he did it was he went ahead and he got an animal that had anthrax, and he took some of its blood, and he went ahead and he injected the blood into a healthy animal with that infected blood. And sure enough, the healthy animal did get sick with anthrax. And now how did he prove, well, okay, that it's the same bacteria? He went ahead and he took a sample from the second animal that he injected, and he looked at it under the microscope, and it was the same bacteria. So he was able to discover that a certain bacteria can lead to a specific disease. And that was, again, a very important discovery during that time. Edward Jenner made a huge discovery. He discovered the concept of vaccination. Now, how did he discover how to even make vaccines? Or not how to make vaccines, but the idea that there, we could get vaccinated, we could build immunity against a specific disease. He came up with that idea. So what he did was he got someone infected with cowpox. We don't hear of cowpox now, but back then there was cowpox. So he basically collected some of the scrapings, okay, from a blister from someone who had cowpox. And he went ahead and he took those scrapings and he injected a volunteer. Now, we totally don't do that now, but back then they did. So he went ahead and he injected um, a volunteer with the cowpox, and the volunteer was actually an 8-year-old girl. So what he did was he scratched her arm and he inoculated her with the cowpox disease. And then, of course, she did get sick. And so he discovered that, okay, the girl did get sick, but then the girl recovered. And after she recovered, he inoculated her a second time, and he found out that she didn't get infected the second time. And so he discovered that she actually built some immunity. And she was also infected with smallpox. So also being infected with smallpox, which is pretty similar to cowpox, might have also given her, right, an advantage. And she was able to go ahead and get better pretty, pretty quickly. And so the idea of vaccination came to him that if we get exposed to a microorganism, we are going to start building immunity against it so that the next time we go ahead and we see it, we're going to be ready for it. And that's the whole idea behind why we get vaccinated, so that we could be ready for, that microorganism that's going to hit us. And that was an important, discovery right there. Let's talk about Alexander Fleming. So Alexander Fleming, he also made a huge discovery, and that was the idea behind penicillin. And so Alexander Fleming actually discovered the first antibiotic ever, and it was all due to accident. So he actually had a plate with some bacteria growing on it, and he was about to throw it out because there was mold growing on it. And then he discovered an area where the mold was growing. The bacteria actually died. So right here, he has, here's his plate, here's the Petri plate, and he has the bacteria growing on his Petri dish. And then he discovered that right here, when the penicillin started growing, do you guys see how the amount of bacteria is decreasing compared to right here, how clustered they are? So he discovered basically the first antibiotic, penicillin. And so penicillin is an antibiotic from the fungi, from the mold Penicillium. And that was a huge discovery, because then they used penicillin a whole lot. And so that was, again, another important discovery right there. And this plate, by the way, has a bunch of Staphylococcus aureus growing on it. These are some terms that we're going to face throughout the semester. Whenever you guys see ology, the suffix ology means study of. So mycology is the study of fungi, bacteriology, study of bacteria, parasitology, that's when you're studying all about parasitic worms and basically parasites. That's an image of a parasitic worm being pulled out of someone's leg, and actually they say this is how the symbol of the medical profession came about. It was basically like a rod, and they were pulling the worm out of someone's leg, and it was super long, and so they started coiling it around that rod, and so that's how they came up with that symbol right there. Rebecca Lansfield is also a pretty important scientist to talk about, important microbiologist. She made an important discovery. She was the one that discovered that there are some differences in the chemical components of the cell walls of bacteria. So, as I said, bacteria, they have a cell wall made out of peptidoglycan, but she discovered that there are some other components, and these components make bacteria unique from each other. So she looked at a group of bacteria known as streptococci or streptococci, and she discovered that not all the streptococci bacteria out there are the same. They have different components in their cell wall. Virology, that's when you're studying viruses, and the way viruses are grouped, as I said, they're neither eukaryotic nor prokaryotic. Viruses, they are extremely small, so you can't use the microscopes that we usually use in lab to view viruses. The microscopes we usually use in lab are only used to view things like bacteria, of course, things like amoeba, so protists. The microscopes in labs can go ahead and view those. But to view viruses, that's a very special microscope, and those are super expensive. So we're never able to view viruses in the lab. Microbes can also be used to clean up lots of pollutants such as oil spills, and they are able to degrade organic matter in the sewage. So there is definitely so many usage for microbes, and especially bacteria out there. Um, in this class, we are going to talk about bacterial genetics, and we're going to talk about how they can actually pass their genes to each other. We're also going to talk about recombinant DNA, so the concept that I was talking about earlier, how you can take basically DNA from two different sources and combine them together. We're also going to discuss microbial ecology. So we're going to talk about how some bacteria out there can actually produce oxygen. So a lot of the oxygen that we are actually breathing is actually being produced by bacteria. So they're pretty important. So imagine life without bacteria. I don't think it would be possible. They are super important. We talked about microbiota, right? The group of microorganisms that you might have living inside of you, for example, in your gut. Now, inside of your gut, you actually have both good and bad bacteria. As long as the good bacteria, they are the ones in high numbers and they are the ones in charge, you are not going to fall ill. But if your microbiota experiences some imbalance, then you are going to start falling sick. An example would be when someone takes too much antibiotics. Antibiotics can actually mess up your microbiota. Antibiotics can actually start killing your good bacteria. One symptom, for example, is if someone takes lots of antibiotics, they might start feeling cramps, abdominal cramps. They might get bloated pretty fast after, after anything they eat. Okay, their stomach would be bloated, and so that could be an indication that there is an imbalance in their microbiota. And sometimes what physicians would suggest is these individuals should start taking more probiotics when they take an antibiotic. The probiotic would actually, kind of balance what's going on there. We're also going to talk about biofilms in this class. So biofilms, that's when microbes attach themselves to a surface and they grow in masses. And basically, biofilms, you can find them everywhere, even on your teeth. So when you guys floss and you find that white stuff that's coming out, that's basically bacteria. That's bacteria that was stuck on your tooth. And if they actually get stuck on your tooth for a while, let's say due to you not flossing a whole lot, you have to go to the dentist and they have to scrape it off, right? So when they scrape it off, that's basically biofilm that they are scraping off. You can even find biofilm, like a bunch of bacteria growing even underwater. Um, and biofilms can actually cause infections because you end up with a community of microbes. So a biofilm, you don't just have one type of bacteria, you can actually have multiple types of bacteria and they kind of create that strong community. So it's really hard to actually get rid of a biofilm and they can cause pretty bad infections and they can be resistant to antibiotics. And that's an image right here of biofilm on catheters. Actually, catheters are like the worst things ever. I mean, they are pretty important, but they do cause lots of infections to patients. Almost all patients having a catheter would get an infection, and it's just because that catheter is placed in there for for a period of time, and it's usually like a long period of time, and so the bacteria starts to grow within the catheter and eventually it can creep up into your body and therefore cause an infection.